All right, so we're going to be continuing today's lecture with the cell. We started talking in the previous lecture about the origin and the history of cells. We discussed prokaryotic and eukaryotic cells. Today we're going to be digging in a little bit deeper into the structures that make up the cell. So when we talk about structures that make up the cell, we're talking about the insides of that cell, as well as the surroundings of that. Now, the insides of that cell, we refer to those structures, those machinery that's actually responsible for the functioning of the cell as organelles. Okay, so organelles can be of two types. It could be membranous and non-membranous, and we'll talk about the difference between the two. But when you look over here in this slide, essentially you're looking at all the different structures that you would find within this tissue, within the cell. All right. For example, this is a mitochondria. It produces energy. This is called endoplasmic reticulum. So this and as well as that, this is all endoplasmic reticulum. So this part here where you see all the studs, that's referred to as rough endoplasmic reticulum, whereas this part here that doesn't have these studs on them, which are actually ribosomes, is called a smooth endoplasmic reticulum. So we have a difference between rough and smooth endoplasmic reticulum. As we move along, we talk about the plasma membrane and the functions of the plasma membrane. So the plasma membrane, we're talking about all this over here. Okay, this is what separates the outside from the inside. The plasma membrane, it's acting as a barrier, a physical barrier to protect the contents of that cell from its surrounding. In addition to that, it also regulates what is able to come in and what is able to leave. When is something able to come in? When is something able to leave? How much of something is able to come in? How much of it leaves? All of this regulation is done by the plasma membrane. So that includes ions as well as nutrient entry into and out of the cell. So this plasma membrane, it's made up of a phospholipid bilayer, right? Phospholipid bilayer. So what that means is that it has two parts that kind of face one another. That's where the bilayer comes in, and it's made up of phospholipids. So the head is hydrophilic. In other words, it likes water. Okay. The tail, it's made up of fatty acid. It is hydrophobic, and it faces the inside of the membrane. And we'll look at this in a photograph over here, as you can see. So we have one layer over here and another layer over here. So this is this phospholipid bilayer. Okay, so there's one layer here, and this is facing the outside of the cell's environment, and this is the inside of the cell. So you have another layer facing inside. So these two tails, they face one another, and the heads, they're facing the aqueous environment, the watery environment. Because remember, they like water, so it's okay for them. This part over here does not like water. So again, that is hydrophobic. The reason it doesn't like water, it's made up of fatty acids. Now, when we're talking about fluids, depending on where the fluid is found, it is named accordingly. So when we're referring to fluid that's found outside of that cell, we call that extracellular fluid. Another term for that would be interstitial fluid. These terms, they're somewhat used interchangeably. Interstitial fluid, as you may recall, it is also tissue fluid. So you can kind of refer to the same thing. Tissue fluid, again, it's a much broader area, whereas extracellular fluid, it would be the fluid that is directly surrounding a cell. And the broader area that it's covering all the cells and farther away from the cells we found within the entire tissue would be that interstitial fluid. Inside of the cell, we call that intracellular fluid. Okay, so inter is between, Okay, intra, intra means within. Okay, so intracellular fluid, that's within the cell, intracellular fluid. Now, as far as the cytoplasm goes, the cytoplasm is everything you find inside the cell and outside of the nucleus. This is what the cytoplasm is. This is that area. Okay, everything you find inside that cell, the area inside that cell and outside the nucleus would be referred to as the cytoplasm. Now, the fluid that all that within the cytoplasm is called the cytosol. So again, the entire area we said is called the cytoplasm, and the fluid that we find within the cytoplasm is called cytosol, or again, another term for this is that, as I mentioned in the previous slide, intracellular fluid. So within the cytoplasm and within the cytosol, we have a lot of materials that we find. So these dissolved materials within this fluid would be, possibly they could be nutrients, they could be proteins or ions. In addition to that, it's all the products that the cell needs to, the building blocks that the cell needs to produce whatever it is trying to produce. And in that process of producing, it will produce waste. So everything is going to be found within this cytosol. Okay, all these dissolved materials will be found. Also, we find, aside from that, when we're talking about ions, so you'll have potassium, sodium. So generally speaking, you'll also have proteins. Okay, a lot of proteins you'll find over there. Carbs you'll find there as well. Amino acids and fat also they will be found as well. Again, remember, these will be the building blocks, so you won't have too many of them because they'll get converted into proteins. So within the cytoplasm, all right, we talked about what the cytosol consists of. Okay, what are all the dissolved substances that we find within the cytosol? So again, the carbs, amino acids, the proteins, the potassium, the ions, etc., etc., etc. So all these things, essentially, they're there, or they're being produced by, or they're being used by, or they're a result of organelles. Okay, so organelles, these are the specific structures that, or think of them as the mini factories within this huge mega factory, which you can kind of think of that as a cell. So think of the cell as a mega factory, and within this mega factory, you have these smaller factories over there. One of the factories, or one of the areas within this huge mega factory, they're producing energy, so that would be the mitochondria. Another one is producing proteins. Okay, another one is sorting out and distributing the what has been produced. So again, you have specific areas, specific machinery that are carrying out special functions, and that is what organelles essentially are. The organelles, when you look at the word itself, organelle, this suffix E-L-L-E means small. Organ, as you know, means tool. So we're talking about small tool, or small tool, since this is a plural form, organelles. Now, these organelles, these small tools, they can be categorized as either as being either membrane bound or non-membrane bound. So you have what's called membranous organelles as well as non-membranous organelles. So the non-membranous organelles, as the name implies, does not have a membrane. There's no membrane there, and it comes into direct contact with the cytosol. And these include structures such as the cytoskeleton, microvilli, the cilia, the centrioles. You find proteasomes, ribosomes. All the non-membranous organelles, they're in contact with all of this, and they include all these functions. Moving along to the membranous organelles. The membranous organelles, they include so again, membranous organelles, they, as the name implies, it means this is an organelle that is surrounded by a membrane, a plasma membrane. And they are isolated from the cytosol. Okay, so they're not in direct contact with the cytosol. The membrane is in direct contact with the cytosol, Whereas in the non-membranous organelle, remember what we said, it is in direct contact with the cytosol. And when we talk about what are some of these organelles, okay, that are membranous, they would include the mitochondria, they include peroxisomes, they include the Golgi apparatus, they include the endoplasmic reticulum, and even also the lysosomes. They are all membrane-bound organelles. Now, let's look at some of these non-membranous organelles first. So we have six that we're going to be talking about in this lecture. And we'll look at each one of these in detail. So again, remember, non-membranous organelles, they're not surrounded by a membrane. They are in direct contact with the cytosol. And we're going to be looking at the cytoskeleton, microvilli, centrioles, cilia, ribosomes, and proteasomes. So in terms of cytoskeleton, so cytoskeleton, this is a structure that helps the cell maintain their shape in addition to its internal organization. In addition to that, it also provides mechanical support that's going to allow cells to carry out essential functions like movement as well as division, cell division. Now let's look at some of these specific or some of the components of that. So again, cytoskeleton, this is a structure that helps the cell maintain their shape in addition to its internal organization, and it's going to help it carry out a lot of functions. So the cytoskeleton is made up of multiple components, and some of these multiple components that it consists of include microfilaments, intermediate filaments, as well as microtubules. Microfilaments, they're also called, sometimes they're referred to as actin filaments. And if you guys recall from your anatomy, or perhaps even, from your anatomy, muscle, skeletal muscle, we have what? Myosin and actin, okay? So this is the same actin over here. Actin filaments, okay? Now remember, these are protein filaments that we have. We find this in eukaryotic cells, okay? So in eucs, we find microfilaments. So it ends up forming part of the cytoskeleton. In addition to that, its functions, they also include cytokinesis, cell motility. It can help the cell change in shape, so endocytosis, exocytosis, and as I mentioned, in skeletal muscle, when they are part of that, again, what do? Their job is for contraction, cell contractility. The next type, intermediate filaments, they provide structural support, and they also regulate key signaling pathways. Now, microtubules, on the other hand, along with intermediate filaments and microfilaments, these are the parts of the cell's skeleton that'll determine the shape of that cell. And they're involved in different functions that include assembly of the mitotic spindle during cell division. You can also find them as axon extensions in neurons, nerve cells as well. So again, when we're looking at microfilaments, we also call these actin filaments, and remember, they're found in eucs that end up forming part of their cytoskeleton. And in terms of its functions, remember what I tell you, for cell motility, it helps change the shape of that cell, activities of endocytosis and exocytosis. Actin, as you know, is also found in muscle, therefore it has cell contractility function, in addition to providing mechanical stability. Remember, in skeletal muscle, we find what? Actin and myosin working together for muscle contraction. The next one, when you spoke about microtubules, these are large hollow tubes of the protein called tubulin. Okay, so tubulin or tubulin, You can pronounce this however you like. This is the protein that forms microtubules. And along with intermediate filaments and microfilaments, they're part of the cell skeleton. And this is what helps determine the overall shape of a cell. In addition to that, microtubules, they're also involved in functions that include cell division. Okay, so this is important. And as I said earlier, again, you find them being part of the axon extension within nerve cells, within neurons. When we look at microvilli, what they end up doing is they end up increasing the surface area of a cell, and that aids in the job of absorption. Microvilli you can also attach to the cytoskeleton. Now, moving along with centrosomes or centrioles in the centrosome. So, centrioles, they form the spindle apparatus during cell division. So we will find centrosomes in animal cells, and as well we see them in motile algae. So it's made up of a centrosome. First of all, you have to understand it's made up of two individual pieces, okay, called centriole. So when you have two centrioles and they come together at 90-degree angles with one another, we call them now it's called a centrosome. Okay, so what are the building blocks for a centrosome? Centriole. Okay, how many centrioles? Two of them. Okay, at right angles to one another, 90-degree angles. And centrioles, they're made up of microtubules. Cilia, on the other hand, these are small hair-like extensions, and what they do is they help move things across the surface of a cell. Okay, that's their job, cilia. So if you look at this picture over here, essentially you're looking at microvilli. So imagine when you look at your intestines, your small intestines have lots of microvilli. And what ends up with these extensions they do is, imagine if this was flat, okay, if these extensions were not here. If this is all flat, then as nutrient is moving across, whatever that you may have eaten, it'll just kind of go across, okay. Now you have this area that's going across. However, within this area that goes across, you have lots of projections that are going upwards. So now nutrients, they have all this extra area to attach and stick onto and then get absorbed. So again, the job of these microvilli is to help increase absorption. Okay, they help increase the surface area, and that increase in surface area aids in absorption as well. Next, we will talk about ribosomes. Ribosome, you can think of them as the protein factories of the cell, and we find them in the cytoplasm. You also find ribosomes in the mitochondria. In plant cells, you'll find them in chloroplast, and also you can find them attached to a rough endoplasmic reticulum, as well as the nuclear membrane. And ribosomes, they're made up of rRNA, okay? That's what they're made up of, in addition to proteins. So prokaryotes, as you'll learn later on in the course, they have 70S ribosomes. In eukaryotes, we have 80S ribosomes. Okay? So again, the specific type of ribosomes we find in procs are 70S, and in eucs are 80S, 80S, small s. Functionally, these are the sites where protein synthesis occurs. Okay? This is very important. Ribosomes, they could be either free or they could be fixed. If they're free ribosomes, that means they're floating around within the cytoplasm. Fixed ribosomes, they're attached to the endoplasmic reticulum. Okay? And again, both of them, they're responsible for producing proteins. When it is attached to the ribosome, those proteins that get produced, they will be secreted out of the cell. When they are found free floating within the cytoplasm, they are going to be used by the cell, okay, manufacture proteins for the cell. Proteasomes, okay, these are enzymes that are able to break up proteins, okay. So proteasomes break up, they clean up protein, damaged proteins, they recycle proteins, right? That's their job. Proteasomes, they'll take a protein and they'll break them down. What are they breaking down into? Amino acids. So when you look over here, this is a ribosome, and again, very simplistically, you'll see that you have these two pieces, one piece on top, a larger subunit, and a smaller subunit. We'll go into detail later on in the lecture. So again, small subunit, and then also this is the large subunit. So the genetic information, mRNA, it comes through, it gets fed over here, and it gets read. As this information is being read, there's a couple of sites. If you look over here, there's an A site as well as a P site. So as the genetic information is being read, amino acids are being added on from one site and being moved over to the other side. Okay, so the reading gets done over here, amino acid gets attached, then it gets pushed along. Another one, another information, another codon gets read, amino acid gets brought in, and it gets transferred over to the P site, where it adds on to the next, the amino acid before then. And this happens over and over again until this entire mRNA is read, and you get to a part of this mRNA that says stop. It means enough is, we are not adding on more amino acids. So this essentially is how ribosomes will add, again, this is very simplistically, we'll go into detail, but this is how proteins end up being produced. Protein synthesis takes place on ribosomes. All right. Now, so what we discussed in the previous slides were the non-membranous organelles. Now we're going to be looking at the membranous organelles. So we're going to be looking at the endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and as well as the mitochondria. So again, very simplistically, endoplasmic reticulum, it could be either rough or smooth. All right. So endo means within, okay? Endoplasm, okay? Within the cytoplasm. Endoplasmic means pertaining to, okay? And reticulum means network, okay? So the network that's within the cytoplasm, okay? Endoplasmic reticulum. This is what this word gets broken up into. Now, within this endoplasmic reticulum, within the ER, we have these storage chambers, which are referred to as the cisternae. So what is the job for the endoplasmic reticulum? So again, as I told you before, this is where proteins are being produced that are going to be exported out of the cell, that are going to be secreted. Synthesis of proteins as well as carbs as well as lipids. Okay. Now, I said protein before. You guys might be thinking, what about these carbs and lipids? Where are they coming from? Well, endoplasmic reticulum, it could either have these ribosomes on it, or it cannot have the ribosomes on it. Where you find those ribosomes, then they're going to be producing proteins. When the ribosomes are not there, then they produce the other stuff. Of course, when you produce it, you're going to store some of it as well. So storage of some of these synthesized molecules and materials also occurs within the endoplasmic reticulum. And then you're going to be transporting material within this ER as well. Endoplasmic reticulum also has the capacity of detoxifying toxins as well as drugs. Okay, so again, remember, endoplasmic reticulum, it could be either rough or it could be smooth. And what's different between rough and smooth is either the presence or absence of ribosomes. Okay. So when you look at this photograph, or in this, not photograph, this picture over here, this drawing, what we see over here is endoplasmic reticulum. All of this is endoplasmic reticulum. The parts that are studded, where you see these big blue dots, or these small blue dots, these are ribosomes. So if it has these ribosomes attached to it, we call it rough endoplasmic reticulum. All this part over here, where there are not any ribosomes, we call this smooth endoplasmic reticulum. Okay, so you have smooth and rough endoplasmic reticulum. Okay, remember we said we have these storage or these pouches, these storage areas. Here they are over here. Okay, within these folds, these are the cisternae that we see over here. All right. So remember, rough ER, smooth ER. So rough ER, what's being produced? Proteins. Okay, don't forget proteins are being produced there. Smooth ER, what's being produced there? You get everything else, right? The carbs, the lipids. Again, it's going to be detoxifying toxins and poisons. In addition to, it also produces hormones, steroid hormones. So again, these are important things to remember. Now, Golgi apparatus, this is part, think of this, the things that get produced in the ER, they end up getting shipped over to the Golgi apparatus. They consist of, again, they have compartment-like the cisternae that we saw in the ER. And these cisternae, they produce a network of tubules from the periphery. And these tubules, they end in spherical, enzyme-filled vesicles. Think of this as well, you know, in Turkey, I don't think they have that. Depending on which part of the world that you're from, you may be familiar with packaging centers. So from where I'm from in Chicago, they would have these shops. They're called UPS stores, and even FedEx. They had these FedEx shops. You'd go over there, and whatever you have, like, you know, maybe you want to send, I don't know, a gift to your friend that's living in a different city or wherever. You take over a cup, for example, a coffee cup, and you would give it to them. They would wrap it up package it in some styrofoam wrap, and then they would put bubble packaging, and then they'd take that bubble packaging and they would put it into a box, and they would make sure it's nicely secured. Then that box would end up getting taped, and then, you know, they would print up a label and attach to the box to let it know where it's going to be shipped to, and then it gets, you know, another truck would come pick it up, and then it would go out on its way. So think of this Golgi as more or less kind of like doing something like that. It has a similar role in that it's going to be receiving products, it's going to be modifying it, it's going to be packaging it, and then it's going to be shipping it out. It's going to be sending it out. So it's going to be storing some products that it's received from the endoplasmic reticulum. Also the Golgi apparatus, they also produce lysosomes. They end up secreting enzymes as well as hormones, and also they'll end up secreting some of the substances, the cell wall, some materials that the cell wall will need that it uses. So again, this is a very important structure. Okay, so when you look over here you see these cisternae, and you can see that here, these are these secretory vesicles that, you know, as you go away from it. So here this is the endoplasmic reticulum. So materials end up coming in over here, they'll enter here, and then they end up ending up into these secretory vesicles, which then end up, as the name implies, secreted away from this endoplasmic reticulum. The products, they end up getting packaged up like that, they get pinched off, and then they get taken away. Lysosomes, okay? So we talked about proteasomes, and what did we say? Proteasomes, they end up breaking up protein. Lysosomes, these are very powerful enzymes that end up breaking up everything else. Think of this as, so first of all, this term lysosome, lyso means dissolve, soma means body. So these are dissolving bodies, if you want to look at that literally, in terms of what the word is made up of. As I said in the previous slide, they get produced by the Golgi apparatus. This would be for primary lysosomes. Primary liposomes, they would be produced by the Golgi apparatus, in addition to inactive enzymes. Now, these secondary lysosomes, once they're formed, the secondary lysosomes, they will fuse with organelles that are damaged, and then they'll degrade them, they'll break them down. They act as digestive enzymes also when they're activated, and they also have the ability to isolate materials that are toxic. So toxic chemicals, they can also be isolated by them. So again, they will also so these things that we talked about, that I spoke about, essentially what they're doing is they're performing the function of intracellular digestion. They end up destroying old and also non-functional cells. Lysosomes actually end up, so when a cell is about to, let's say the cell gets damaged, right? So there's a mutation that may have occurred. And if the cell is able to recognize that mutation occurred, one of the things that the cell will do is it'll just it'll kill itself, okay? It commits suicide. And that is called apoptosis, okay? apoptosis, apoptosis, programmed cell death, apoptosis. And it's these lysosomes that end up, apoptosis results from these lysosomes opening up, and they just end up pretty much killing everything that's inside, destroying everything, all the organelles inside that cell. So if all the organelles inside the cell end up getting destroyed, then the cell can no longer function, right? And the cell dies. And this is what happens if a mutation occurs or if something goes wrong. This will take place if the cell is too old again, or if it's not functioning, it's not working properly. This will occur. So think of it as like, recycling worn out cells. And so remember, we spoke about these two lysosomes, primary and secondary lysosomes. All right, we'll leave it at that. In reality, there's actually, that I'm aware of, there's actually 4 types. There's primary, secondary, residual, and also autolysosomes. So again, suicidal bags of cells. Okay, it's another term that they give, suicidal bags of cells. So why does this term come from? As I suggested to you, apoptosis. If the cell gets damaged, these vesicles end up getting released. They have the capacity to kill this entire cell. Also, again, they destroy it. They're very powerful enzymes. They destroy lots of things. When this whole COVID started, one of the things that people were doing is they were using lysosol they were spraying the surface areas and stuff, right, to destroy the virus, etc., etc. Lysosol was the name of this product. Again, it's sold in the United States. I'm not sure if it's available elsewhere outside of the U.S. But again, when you look at it, why is lysosol so good at killing all these germs? Well, because it consists of lysosomes. It has special type of chemicals that are somewhat similar. That's where the name came from as well. All right, so we spoke about lysosomes. We talk about its functions. Okay, autolysis, we spoke about that also. It ends up, so it can clean up things inside the cell, break down molecules. It can even attack, so if you look over here, food particles as they enter some of these lysosomes, they end up helping break down some of these ingested particles. Lysosomes can also go and attack bacteria and destroy bacteria. So again, remember what I said about apoptosis, programmed cell death. So when you look at this term autolysis, or this word it ends up, when you break down this word autolysis, it consists of auto, which means self, and lyse, which means break. So self-breaking, autolysis. So how is this cell destruction, the self-destruction of a cell takes place, of an old cell or damaged cell? So that membrane, remember, lysosomes, they're membrane bound, right? So inside this membrane, you have these very powerful digestive chemicals that are there. So that membrane ends up getting broken down, okay? The membrane degrades. As that membrane degrades, and all these very powerful digestive enzymes, they end up being released. As they get released, guess what? It starts to break everything inside that cell, okay? It'll start to decompose that cell. And then once everything gets broken down in that cell, whatever those materials are, they end up getting recycled, they get reused. So it's rather efficient as well. You don't really end up wasting, okay, but it gets recycled. Your body is probably, well, for me at least, it's the most efficient machine that I am aware of, okay, on the face of this planet. This is how God has designed. God designed everything to be conservative in nature, not to waste. And again, it starts all the way at the cellular level as well. Moving on to the mitochondria. Mitochondria, they could be spherical or they could be rod-shaped. It's a double membrane, so it's got two membranes that are there. It has an outer membrane that's smooth and an inner membrane that produces these finger-like foldings that we call the cristae. The inner membrane, it also has these stalk particles that are called Racker's particles or oxysomes. And the mitochondria ends up being filled with granular mitochondrial matrix. Okay. This matrix, it has circular mitochondrial DNA, RNA, 70S ribosomes, proteins, enzymes, as well as lipids that are found over here. So the mitochondria, it's the only organelle that has the capacity to reproduce itself, okay, all on its own. Okay, why? Because mitochondria has its own DNA. All right. And what is a mitochondria? What's the main job of it? Well, mitochondria takes an energy source such as sugar or carb, and it will end up producing that as it'll convert it into ATP, adenosine triphosphate. Okay, so mitochondria will produce energy, ATP. It'll use glucose and convert it into adenosine triphosphate. And adenosine triphosphate is what the body needs to do work. This is the energy, this is the fuel for the cells to function. So for going back to the mitochondria, continuing with the mitochondria, the energy production process in the mitochondria can occur in one of two ways. It could be either aerobic or it could be anaerobic. So again, going back, remember, mitochondria is producing adenosine triphosphate or ATP. So again, these are very high energy rich molecules. Okay, so when this takes place aerobically, so again, what's the difference between aerobic and anaerobic? Aerobic essentially means with oxygen, anaerobic means without oxygen. If oxygen is present, okay, the mitochondria will take a source, an organic source, such as, for example, glucose. It'll take glucose, and in the presence of oxygen, it'll convert it. It'll carry out a series of chemical reactions, and the first one is referred to as glycolysis. During glycolysis, 2 ATP gets produced, and an end product. That end product, which is pyruvate, ends up going into Krebs cycle, ends up producing another 2 ATP, and a couple of other end products. Those 2 other end products ends up going to another series of chemical reactions, which are called the electron transport, and that will yield another 32, maybe 30 to 34 ATP molecules. So in total, you gain anywhere from 36 to maybe 39 ATP molecules in aerobic cellular respiration, okay, aerobic respiration. In anaerobic respiration, this is occurring without oxygen. Therefore, you're not going to end up producing as much ATP. Anaerobic respiration will yield only 2 ATP molecules. Okay, so this is the difference between aerobic and anaerobic respiration. Okay, so aerobic respiration, it's a series of metabolic reactions that takes place in the presence of oxygen, and it's taking another substance, chemical substance, and converting it into adenosine triphosphate in the presence of oxygen. Whereas anaerobic respiration, again, it's another process that'll take a energy source and convert it into ATP without the presence of oxygen. Okay. All right. So when you look at this photograph over here, what are we seeing? You're seeing the picture of a mitochondria, and remember what we said. It's a double layer membrane. So you have this outer membrane, and then here's that inner membrane. And these inner membranes, they have these. So you have this intra membrane over here, And then you've got the inner membrane. The intramembrane is between the inner membrane and the outer membrane. You can see genetic material here as well, okay, that you find within the, so what ends up creating is a matrix. So between these folds, this area, we call this, this would be the matrix that are produced over here. Okay? And you see all these ribosomes that are here as well, that you find. These are these 70S ribosomes that I had mentioned to you earlier. So again, within here, all the energy production is taking place. So it's a highly metabolic area of the cell. So the largest part, when you look inside of the cell, the largest and the most prominent area of the cell would be its nucleus. So you can consider that to be, again, so as the control center of the cell. It's surrounded by a membrane. We call that a nuclear membrane. And this is also a double membrane. Okay, yeah, it's a double membrane structure. This nuclear envelope. Between the two layers, we have a space, and we call that the perinuclear space. And within this membrane, there's also openings or holes or passageways, and we call these nuclear pores. So if you look over here, this is that double membrane, and within this double, can we zoom in? If you zoom in over here, you can see here 1, 2, and these are this nuclear membrane. Here's that the perinuclear space would be over here. This part that's lighter in color. So you have the inner membrane, okay, and this is the outer part, okay, and then in between you have this perinuclear space. And then over here, these are, or here, you can see another one over here, another one over there. This is what this is, because remember, this is it's a cut, it's a cross section. So what you're looking at here, these are these nuclear pores. So, and it allows for things to move in and out, a passageway between the cells. Okay, so we can kind of zoom back out over here, and as we zoom out, yeah, so what do you see? And then you see this very dense area. We call that the nucleolus over here. And all this is just chromatin, okay, that you have that's floating around within this membrane-bound structure, okay? And this entire area we call, the entire area inside this nucleus is referred to as the nucleoplasm, okay? Nucleoplasm is the entire area within the nucleus. So, moving on to the nucleoli. All right, so now let's look at, all right, so we talked about the nucleus, we talked about the membrane. So remember, all the genetic material is stored inside the nucleus. So the nucleoli, this is the area inside the nucleus of that cell that's made up of ribosome, well, RNA, enriched in proteins, and this is where RNA is going to be produced. Ribosomes, again, these are the products, these are the tools that are going to be bringing amino acids together. They're going to be connecting amino acids to form proteins, right? So these are the protein making machinery of the cell, okay, of the nucleus. When you talk about nucleosomes, this is a structural unit that we find in eucs, okay, in eukaryotic chromosomes, and it's made up of a length of DNA that's coiled around a core of proteins called histones. And we're going to be looking at this in detail later on in the course. So what is chromatin? Chromatin is, again, so we're continuing with looking at the contents that we find inside the nucleus. And chromatin is just loosely coiled DNA. And we find this in cells that are not dividing, okay? When cells are about to divide, then that DNA is not loosely coiled anymore, okay? It's super condensed. It's very, very tightly coiled. And we call that chromosome, okay? So tightly coiled or super condensed DNA is referred to as chromosome. And this only occurs when the cell is dividing. So, as I had mentioned, the nucleus of a non-dividing cell, it will have a network of these fibers that we called chromatin. Okay? During cell division, that chromatin, it's going to condense to form these very distinct chromosomes. So if you look at human DNA, you see all these little X-shaped structures. That's just very tightly supercoiled condensed chromatin, okay, that ends up forming these chromosomes. Chromosomes, they help in the transmission of characters or traits, okay, or genes. And chromosomes, they have, it consists of a centromere at the center, and it's got arms on either side of the center. Okay? We call these the chromatids. And chromatids, you could, again, think of these as these thread-like when you look at the picture, you kind of get a better idea of these. So chromatids, thread-like chromonema. So when you look over here, what do we see? This is a typical chromosome. And again, what is it made up of? It's made up of chromatin. Okay, that's been super condensed, very tightly coiled around histone proteins. And you can see this, the center part, this is the centromere. And then you can see that there's, first of all, there's two of these, one over here, one over here. See, these are the sister chromatids. So the short arm that you see over here, this part up over here, this would be the P arm. And the longer one over here, this would be the Q arm. So you have a P and a Q arm that's of this chromosome that we have in this picture at least. This may not always be the case, and you guys will learn about this later on in the course when you look at this into more detail. Again, this is just very general. It's a very simple overview. We will be going into a lot more detail of this as the course progresses. This is just kind of opening the doors and giving you a little bit of exposure as to what's to come. End this lecture, this topic, with this quote: cells regarded as the true biological atom. So remember, what are atoms? We said that atoms are the building blocks of matter, right? So if you want to look at the building blocks of life, then you can regard the cell as the true biological atom. Right? Hopefully this makes sense. Think about this, percolate on this. All right, so that's it for this lecture. Thank you, and we'll see you guys in class.